Yeah, good, and nothing else.
Because yellow is the dominant recessive trait.
Little g is yellow.
Yellow.
And yellow is recessive, and the only time we see a recessive allele is when we don't have a dominant allele.
So tell me the combinations for green pea pods.
Big G, big G.
What's that?
Big G, big G.
Big G, big G. Any more?
Big G, little G.
Good. Any more? No.
Okay, so these are important vocab terms that from here, hopefully we can, this is kind of the groundwork, right? This is the key terms that if because I should be able to say doing a Punnett square, we have a yellow pea pod is crossed with a heterozygous. And those words are enough that, hey, it's a yellow pea pod, little g, crossed with a heterozygous, that would be crossed with a big G, little g. Those are words that we should be able to just say because you know these terms and they can go through and apply. Now these are completely dominant traits and we'll kind of transition away from those today, but that's what we're gonna deal with. So let's take a look at question number 1 on that half sheet. Okay, so again, we went order of number 3 and we're gonna do number 1 and number 2. So these are dominant traits, these are completely dominant traits. And we are telling you that dimples are dominant over no dimples. Now, we don't give you a letter to use. You could use any letter you want. Just realize that a dominant letter needs to be a capital. Okay? Typically, we get lazy and we just use the letter that the word starts with. Okay? We don't always, but it works kind of nice. So if we choose D for our dimples, then that's what. If you use the letter R, then it'd be a big R. It doesn't really matter, right? As long as you know that dominant is capital. So a man who is heterozygous, okay, so what would their genotype be then? Big D, little d. Has a child with a woman with no dimples. So what was mom's genotype? Little d, right? So dad is heterozygous, mom is homozygous recessive, because she has the recessive trait, and the only time we see the recessive trait is when it's homozygous recessive. Is it possible, now the question that I'm asking you ultimately, is it possible for them to have a homozygous for dimples? Now is that big D, little d, or little d? What question am I asking? Homozygous for dimples would be big D, right? So is it possible, well, here's the Punnett square, and I guess maybe let's make sure we're solid on the genotypic and phenotypic ratios, because this is pretty like standard procedure when we do Punnett squares, or what are the possible outcomes. So out of my 4 boxes, 2 are going to be big D, little d, and 2 out of my 4 are going to be little d, which means the dominant trait is being expressed because it's a big D. So dimples and dimples, no dimples, no dimples. So is it possible to have a big D child given these 2 parents? No, right? It's not possible for a child to be homozygous dominant. It's not possible for these 2 parents to produce a big D child. Questions on that one before we flip the page? Which one are we just do together? You don't know? Why are you? Oh, then should I take it? Because okay, we just did number 1. So number 2, dimples are a dominant trait, and a man has no dimples, and they have a child with a woman who has no dimples. So man with dimples and a woman with no dimples. What must mom's genotype be? What must mom's genotype be if she does not have dimples? Little d. And what's dad's genotype? Big D something. Let's just leave it at that. It's big D something, right? For sure he has a big D. We don't necessarily know what it is yet. If the child doesn't have dimples, let's start there. What's the genotype of the child then? Little d, right? The recessive trait is only seen when it's homozygous recessive. what would the genotype of dad have to be then in order to produce a child that's little d, little d? What's mom gonna pass on?
A little bit.
Or,
A little bit.
okay, so that one came from mom. Sure. Where'd that one come from?
Dad.
So what does dad's second letter have to be? Has to be a little d. Dad has to be heterozygous, right? And we can do the Punnett square really quick to show you that if dad is big D, little d, we could get a little d, or little d, little d. However, if dad was big D, would they ever produce a little d child?
Nope.
No, right? Because they're either dads would pass on a big D or a big D and only a big D. So that kind of shows that it doesn't work. Verifications on completely dominant traits before we move on. All right, well then, what was previewed in your section 3 guided notes was this idea that sometimes traits, either the sheet or a sheet that you need to grab a packet. Not all traits are completely dominant. Sometimes we have some other modes. Now, we are going to talk about today something called incomplete dominance. We're going to talk about something called codominance, and we're going to talk about something called sex-linked recessive. So there's 3 other modes of inheritance that we're going to talk about today. Now, these 3 modes of inheritance also don't talk about something that was referenced in your notes called polygenic. And the idea is a lot of our traits are not controlled by a single gene. There's a combination of lots of different genes, many poly, that combine to make you look what you look like. So why is your hair color the color it is? There's multiple genes that are put together to give you the hair color you have. I believe eye color is also the same. I believe skin color, I think, is also like that. Okay? So remember back to when we talked about Mendel. Prior to Mendel, there was this idea of something called a blending hypothesis. Because there are certain species, a snapdragon is an example of a flower, that if you take a red flower and you cross it with a white flower, we get a pink flower. So what is a pink? Well, it's somewhere in between, and that's how I always remember incomplete. It's somewhere in between red and white, right? Pink is in between those two. So when we talk about incomplete dominance is when the trait is in between the homozygous traits. Incomplete is in between. Now, a blending hypothesis said that if we take a pink and a pink, for here on ever, we will get pink, right? If I take the example was water, blue water and yellow water, I get green water. If I take green water and green water, from here on forever, I'm always gonna get green water back, because they blended together. However, that's not the case when we talk about genetics and incomplete dominance. Blending hypothesis is not correct. Mendel disproved that. So snapdragons are incompletely dominant. So the ideas are very similar. The mechanisms of Punnett squares doesn't change. The genotypic ratios don't change. It's really the interpretation of phenotypes is the only difference that we're talking about here. So we have 3 different phenotypes. We have red flowers, we have pink flowers, and we have white flowers. The phenotypes, what we're physically gonna see are red, white, and pink. 3 phenotypes now. The red flowers are homozygous dominant. So what's gonna be the genotype for a red snapdragon then? Using R's, it says R's. So homozygous dominant, big or littles, big and, what's the second letter? Big R and big R, right? Homozygous, same, homozygous dominant, big R, big R. Where white flowers are homozygous recessive, so what's this genotype gonna be? Little r, and pink is the heterozygous trait, so that means that's gonna be, I'll fill it in for you, big R, little r. The genotypes dictate the phenotypes. Okay, so being able to go kind of back and forth between these is going to be important. So the first Punnett square we ask you to do is to take a pink snapdragon and a pink snapdragon and cross those 2 snapdragons together. So a pink crossed with a pink, what are we going to get? Okay, so big R, little r is our pink genotype, And the other one is also pink, so it's gonna be a big R, little r. The Punnett square mechanisms and filling out this Punnett square is absolutely no different than the Punnett squares we've been doing up until this point with completely dominant. Nothing changes in terms of filling in my Punnett square. Nothing changes when I look at the genotypic ratios. I'm still looking at how many of each letter combination do I have. I have 1 out of 4 big R, little r's. I have 2 out of 4, or 1/2 big R, little r. I have 1 out of 4 little r, little r. Nothing has changed so far. The only thing that changes with incomplete dominance is the phenotype outcomes. My phenotypic ratios are now different. Remember, we have to take this information and we want to cross-reference that chart to determine our phenotypes. So my big R is going to be red. This box is going to produce a red snapdragon. What's this one going to produce? Pink, right? What about this one? Pink. What about this one? White. The only thing that changes, if this would be a completely dominant trait, we would have had 3/4, whatever the dominant trait is, 1/4 recessive trait. We now have something that's in between. So we have 1 dominant trait, red. We have 2 heterozygous, which is the in-between, the pink in this case. And we have 1 recessive trait, which would be the white. We give you a phenotype, you determine genotypes, do a Punnett square, interpret the Punnett square, look at ratios. The mechanisms are the same, the ideas are just our phenotypes, our outcomes are gonna look a little different. What do you think? Another snapdragon or jump pages?
We were pretty tight last
I
night,
know.
so let's go right, let's jump.
Or the other option is we just do 2 snapdragons and no palominos. I don't care.
Let's do Palomino.
Okay, so the rest of the, whatever we don't finish today is your homework, okay? Whatever we don't finish today is your homework. So we did number 1 together, that means on page 1, page number 2, and number 3 are your homework for your virtual base. Continue talking, going through and looking at Snapdragon. On page 2, it's another example of incomplete dominance. Talking about horses, okay? Now, by no means am I gonna claim to know much about breeding horses. Okay, but per this example, we're saying that incomplete dominance is true in horses, that a brown horse is big B, a white horse is little b, and the genotype big B, little b, called heterozygous, will give us something in between. So not brown, not white, but kind of a yellowish tan. It's in between, which then apparently is called a palomino. Okay? So based upon that information, those are the genotypes and phenotypes, those are the connections between our two. If we have a brown horse and we are going to cross it with a white horse, what are the possible outcomes? So again, it references that brown horses are big B. It references that white horses are little b. So the parents are big B, and little b. Right, because I have a brown horse and a white horse. And if we cross a brown horse and a white horse, what we are going to get is 4 out of 4 of our boxes are going to end up being heterozygous. They're going to be a big B, little b. Now, if this was a completely dominant trait, we would see the dominant trait, which would be brown. However, this is not complete dominance. This is called incomplete dominance. And when we talk about incomplete dominance, the heterozygous is in between. So what's in between brown and white? Tan, yellowish, palomino is the idea. Okay? Then we continue on, brown horse and a palomino horse, we would do that cross. We look at a palomino and a palomino, and the idea is you on your own should be able to determine these genotypes given the phenotypes. Are there questions about incomplete dominance before we move on? The only difference between these and complete dominance is that the heterozygous is in between. Punnett square part is the exact same. Questions? Phenotypes, genotypes, but it's really only the heterozygous that changes. The phenotype of the heterozygous is in between. No question? Okay, then there's a new mode of inheritance that we're gonna talk about. It was referenced in your guided notes, just like incomplete dominance was. It's called codominance. And codominance is when we have two, co means like together, like this is co-taught, Mr. DeVore and I co-teach this class, we teach this class together. There are two dominant traits that will both be expressed in the event of a codominant trait. So again, chickens, not claiming to know everything about chickens, but the idea of black chickens are gonna have the genotype of big B, big B. A white chicken is gonna have the genotype of WW. Now notice they're both capital letters, the B's are capital and the W's are capitalized, and the reason is they are both dominant traits. Okay? And what happens when we have something that's called codominance, that when they are codominant is we see both of the dominant traits. Okay? So if we have a black chicken, big B, big B, okay, that's the genotype of the one. We have a white chicken, which is a WW. If we take a black chicken, black, black, big B, and we cross it with a white, WW, what are the outcomes gonna be? So Punnett square looks no different. We still have the B's, we still have the W's that are gonna get filled in. The difference with the codominance is when we have a heterozygous, these can still be said to be heterozygous because their alleles are different from one another. The thing that is different about them is when we talk about the phenotype, they are both dominant, so both traits are going to be expressed. So checkered chicken is what they refer to it as because they have both black and white feathers being shown at the same time. That's a codominant trait. Codominant is when both traits are expressed. Okay? And that's really just kind of moving forward with chickens is the idea. Checkered is the heterozygous, white is homozygous white, and we have homozygous black when we have black chickens. Blood type is another example of something that is referred to as codominant trait. It's also something called multiple allele because there's more than two different varieties. Okay? So when we talk about blood types, well, quick show of hands. How many of you know your blood type? Hey, Mr. DeVore, there's 3 students in this class that know their blood type. That makes 4 of us in this class that know what our blood types are. Last hour, I was the sole person who knew their blood type. Now, honestly, unless you donated blood and you look at your card and be like, oh, I'm this blood type, I understand why you probably don't know what your blood type is. Now, when we talk about blood type today, we are talking about blood types O, A, B, and AB. There's also something called an RH factor that has to do with plus and minus. So you could be O positive or O negative. You could be A positive, A negative, AB positive, right? You have all the combinations. This is only talking about the blood types, not the positives and negatives. So when we talk about blood types, there are 3 different alleles that we talk about when we talk about blood types. There is the A allele, there's a B allele, and there's an O allele. Blood type is a codominant trait, and the A allele and the B allele are both dominant. Okay, A and B both are dominant. O is a recessive allele. Okay, so we have 3 different alleles. We call them multiple alleles because there's more than 2. There's A, there's B, and there's O. A is dominant, B is dominant, O is recessive. So, pretty common question that people ask is like, hey, what can I donate and who can I donate to? If you are blood type O, you are referred to as the universal donor. You can give it to any person because it has to do with these things called antibodies and antigens. Okay, and basically the really quick story is there's proteins in your blood, and if you get the wrong protein in there, your body sees those blood cells as foreign invaders and they attack, which then causes your blood to clot. which is bad. You don't want chunks of blood floating around because it gets stuck. That's bad. Okay? So if you get the wrong blood type, your blood will clot, bad things happen. So what happens is if you're blood type O, you don't have anything that anybody is gonna clot with. So you can give, if you are O, you can give to A, you can give to O, you can give to B, and you can give to AB. You're called a universal donor. Okay? A can give to A or AB because AB blood has both A and B traits there. B can give to B and AB because they have they have B blood type, but B has both A and B. However, if you are AB, you have both A and B blood parts, and any other blood type would see that as a foreign invader. So AB can only give to AB. Now, if you notice, every single line flowed to AB. This is called a universal recipient. They can get blood from absolutely anyone. Ignore pluses and minuses. They can get blood types from anyone. However, if you're blood type O, who can you get blood from? And only O. Okay? O. Okay? And then there's positives and negatives, and the idea if you're negative, you can only get negative. If you're positive, you could get positive or negative blood. But that's kind of called that RH factor. So when we talk about these, and this is the idea, let me just quick. If you're blood type A, you have the A antigens with B antibodies. So basically it sees B's as like bad and invaders, and it will attack anything that has a B in it. But O has both A and B antibodies. So it's gonna attack if you put A blood in there or you put B blood in. Okay. Where AB is not gonna attack anybody. So these are the possible alleles, and this is something you probably wanna write down, is the possible allele combinations that will give you different blood types. Okay, so if you are blood type A, you could be homozygous for AA, or you could be heterozygous with O because O is recessive. So if you're blood type A, you could be AA or AO. If you're B, you could be BB or you could have BO. AB means you have A and a B, and that's the only option you have because you are codominant. You see both the A and the B, and if you are blood type O, the only thing you can be is recessive. You're OO. Okay? So, when we look at these, and it talks about this up top, and I would encourage you to write these down, there are two dominant alleles. A is a dominant allele, and B is a dominant allele, where O is the recessive allele. So if you are homozygous, we still use the same words, homozygous for same. If you're homozygous for B, that means you have a B, and then homo means same, so you're BB. Okay? And if you were blood type, for blood type O, the only time you can have the recessive trait is if when it's recessive, you must be OO. Okay? So if the woman was homozygous for B, and if the man was blood type O, now we don't have to say it, but we could say homozygous for O, it's not necessary because it's a recessive trait, then we can go through and we can fill out a Punnett square. And these Punnett squares are exactly the same as any other Punnett squares. We just have O's and B's, and that's really it. So if a woman who is homozygous for blood type B has a child with a man who is O, we could say homozygous O if we really wanted to, but we don't need to, then 4 out of 4 of their children are gonna have BO.
Oh, no.
Oh, yes.
It's a lot of BO. It's a lot of BO.
It is a lot of BO, okay? Which means they're gonna have what blood type?
B.
B, right? Because B is a dominant or recessive trait. B is dominant. What is O? Dominant or recessive? So what do we see then? The dominant B allele. That O is hidden, that O is hiding. And then it's different combinations of these. But this, I think, again, is gonna be really kind of helpful. Because if I say, and I mean, there's enough information up top, but it says, hey, a woman has blood type AB. What does her genotype have to be? It has to be AB. You know, so there's certain things. Now, if I could say a person like Mr. DeBoer's blood type is A, because that's all a test shows is A. It doesn't say AA, it doesn't say AO, it just says A.
Ayo.
I don't necessarily know what Mr. DeBoer's blood type is just by doing a blood test. We'd have to look at some family lineages and kind of trace those traits through. Okay? So if he was blood type A, there's maybe a question as to which one he is, but based upon some other information, we can maybe figure it out. Okay? Questions on codominance before we move on? All right, then the last mode of inheritance is something called sex-linked. Just to review, sex-linked is tied to your sex chromosomes. Remember, female XX, if there's a Y, there's a guy. If there's a Y, it's a guy, is the male. Okay, so keep that in mind. So there's a certain kind of traits that are carried on the sex chromosomes. Generally, there's carried on the X chromosome. There are a few that are carried on the X, I'm sorry, on the Y, but the ones we tend to talk about, the ones we do talk about, are carried on the X chromosome. So the idea is we can carry these dominant recessive traits on X chromosomes. There is nothing on a Y chromosome. So keep in mind, a dominant trait is expressed if there is a dominant allele. The only time we see a recessive trait is when there is no dominant allele. Okay? So for all these sex-linked traits, the first thing is, what are the sex chromosomes of the individuals? XX, female, XY, male. That's the starting point. Okay? And if we really wanted to, we could build our Punnett square with sex chromosomes and sex chromosomes only. I'm not saying that's the best choice, but we could, right? We could go through and we could do this and we could say, hey, 50% are gonna be male, 50% are gonna be female. Well, let's not do that yet because we gotta go back and add some stuff. But up in the description, it talks about this thing called color blindness. And color blindness is a sex-linked recessive trait, and it's carried on the X chromosome. So this says normal vision is X big B. So if you have you're not colorblind, normal vision, that means you have at least one X big B allele. At least one, because that's a dominant trait and it's going to show the dominant trait. If you are colorblind, that means you are X little b and you don't have an X big B. Okay? There is no X big B in your genotype. So a female that's heterozygous is called a carrier. She doesn't have colorblind, she doesn't have the trait, but she's carrying the trait on. It can get passed on to the next generation is the idea. So a man who is colorblind, okay, colorblind means there is no dominant trait. So he does not have any X big B's. He only has X little b's. Now, how many X's does dad have? And only one. So that X little b is the one and only X that he has. Okay, now what if mom, just for a second, what if mom was colorblind? Does she have any X big B's then? No, she has no X big B's. And how many X chromosomes does she have? So both of those would have to be X little b, just so we're clear on that. Okay, it's the absence of a dominant trait. So mom is homozygous dominant. Okay, homozygous means same. Dominant allele, we're using B's. So what does that make mom? X big B, homozygous, X big B. Is mom colorblind or not colorblind? Not, right? She has normal vision is what we would say. She has normal vision. So these Punnett squares, I would start by doing the sex chromosomes first. And statistically, 50% of these people are going to be males and 50% of the outcomes are going to be females. And then we attach the B's, and they are carried on the X and the X chromosomes only. Notice how we have never written anything on the Y chromosome, because there isn't an allele on the Y chromosome. It's carried on the X chromosomes and the X chromosomes only. Our genotypic ratios are really no different than we've done before. We're counting all possible outcomes. And in this situation, we have 2 out of the 4, or 1/2. For the females are going to be X big B, X little b. And of the males, 2 out of the 4, or 1/2, are going to be X big B, Y. Now the next couple questions are talking, I'm asking percentages, you could do it as a fraction, but I'm specifically asking as fraction, or percentages, is only referencing the females. So yes, we have 4 boxes here, but of these 4 boxes, how many are females? 2, only these 2. So when I look at the question, when I answer this question about the females, I am only looking at the 2 female boxes. Of the females, how many of these females are going to be colorblind? Is this have, remember, if there's a dominant allele, the dominant allele overpowers it. Is there a dominant trait allele here? And what is that dominant trait? Normal or color vision? I'm sorry, normal or colorblind? Normal. So what is this female going to be? Normal or colorblind? Normal, right? That says normal. What about this one? Normal, right? So of this, how many females are going to be colorblind? 0. 0% of the females are going to be colorblind. We only have 1 B to look at the males. There's nothing on the Y. So we only look at the X chromosome. So this X chromosome has a dominant allele, which is for normal color vision. Is this boy going to be colorblind or normal? Normal. What about this one? Normal. So what percent of these males are going to be colorblind? None. 0. Carriers, up in the vocab, the introduction, it talks about being heterozygous. A female that is heterozygous is known as a carrier. So how many females are going to be heterozygous? Of the females, how many are going to be heterozygous? Or are going to be carriers? Out of 2, how many are heterozygous? We're looking at the B's. So are these B's the same or different? Different, and this one is? So this is heterozygous, and this is heterozygous. So 2 out of the 2, or 100%, are going to be heterozygous, which means they are carrier. Let's do number 2 together. A male who is normal, well, why don't you try to write the genotypes. You just write the genotypes for a male who has normal vision, has a child with a female who's heterozygous. Let's just start by you filling in those 4 little spots for next to dad and mom. Start with the X's and Y's first, and then add the appropriate B's where they need to be. Let's start there. I'll give you 10 seconds. Okay, that's 10 seconds. Dad has to be XY. Y, it's a guy. Mom's XX, okay? Dad has normal. So what letter goes on the X? Is it a big B or is it the little b? It's the big B. He has the dominant trait showing through, which is the big B, which for normal color vision. Mom is heterozygous. What are her B's going to be? The same or different? Different, so she's going to have a big and a little b. So it's going to be X big B, X little b. What's another word for heterozygous? Female? What is it? Oh, carrier. It says it up in the introduction that she would also have been considered a carrier. So instead of saying she is a heterozygous female, we could have said she's a carrier for colorblind. So this is our Punnett square. The ones with the stars represent male or female. Female, because it's XX, okay? So when we talk about genotypic ratios, it's a 1 out of 4, 1 out of 4, 1 out of 4, 1 out of 4, right? There's 1/4 this, 1/4 this, 1/4 this, 1/4 this. Each box is a different genetic combination. Each one is a different genotype. Of the 2 boxes that Mr. DeBoer starred to represent the females, of those females, how many females are going to be colorblind? Remember, female has to be little b, little b. So how many of them are going to be colorblind? 0. None of them. No females are going to be colorblind. Of the males, how many will be colorblind? 25. Well, of the males, 50% of the males. Overall, it'd be 25, but if we say of only the males, then 50%. How many of the females will be carriers, which remember another term for carrier is heterozygous, so 50% of the females are gonna be carriers. Now let's just talk really quick. In order for a female to be colorblind, what does she have to be? X little b, X little b. Where did one of those X's come from? One came from dad, and the other X came from mom. Okay? Does mom have to be colorblind in order for the daughter to be colorblind? No, right? She could have been a carrier, right? She could pass on an X little b. Mom could have an X little b without being colorblind. If dad gives an X little b and mom gives an X little b, dad gives an X little b, was dad colorblind?
Yes.
Is it possible that he was not colorblind? He had to have been colorblind, right? Because if dad gives an X little b, what was the other allele that he could have given?
Y.
And did he give the Y? How do we know that? Because it's a daughter, absolutely. Okay, so in order for a girl to be colorblind, dad would have to be colorblind. Just so you know that. Quick show of hands. Raise your hand if you know someone who is colorblind. Okay, look around the room. That's a lot, right? Okay, now put your hands up. Raise your hand if you know someone who is colorblind that is a female. There's some, right? But less. Okay, in order for a female to be colorblind, dad would have to be colorblind, and mom may or may not be. Is anyone in here colorblind? So dad's colorblind. Is your mom colorblind?
No.
No? Okay. Was your grandpa then was colorblind too, I bet, right?
I don't know.
We'd have to trace it through mom, yeah. No, it wouldn't have had to be, never mind. Could have been, yeah.
I'm colorblind as well.
You are? And your mom is, so you're, well, it doesn't matter about your dad. Is your mom colorblind? No. Okay. So, it's less common for a female to be colorblind because she needed to get 2 recessive alleles, and that would have had to have come from dad. Okay? Let's just flip it over and discuss the last one. We're not going to probably do any of these together. The last page is also a sex-linked recessive trait. It's a different genetic trait. It's referred to as hemophilia. So hemophilia is a sex-linked recessive trait. It's carried on the X chromosomes, and it's where your blood doesn't clot like it should. It's a blood disorder. Bleeding disorder, blood disorder. I guess it's probably not a blood disorder. Well, whatever.
Well, yeah, I'll call it.
The idea is if you get cut, you aren't gonna clot like most people would. And clotting stops bleeding, right? So that's the idea. So what for most people would be like, hey, that was just kind of a small, like I skinned my knee. Well, that was kind of annoying, but it stopped. The idea is it keep bleeding and keep bleeding and keep bleeding and keep bleeding. That doesn't go so well. So hemophilia is an example of another sex-linked recessive trait that is carried on the X chromosome. So that's what that problem is looking at variations of a different sex linked trait. So this one we aren't gonna go through, but it's the same mechanisms, but instead of being for colorblind, now it's XH. Big H is normal clotting, where X little h would be for hemophilia. It's still a sex-linked recessive trait, same mechanisms, just different letters, same ideas. So instead of being colorblind, we're gonna say hemophilia. The mechanisms are the same. Okay? All right, so whatever we didn't finish together is your homework for your virtual day.
That's fine.
Okay? So you have until Monday to get this done. And on Monday, if you have any questions on this, we can spend a little bit of time. Actually, we have a warm-up that we're going to go through to kind of practice these. But if between now and Monday you have questions on these, please contact us. Easy way to do it is take a picture, email us the picture, and we can mark it up. And we can send a picture back, and we can kind of address some of those situations. So if you have questions, please let us know. There's a couple of you that still have some tests to make up. So if that's the case, I assigned you on Friday